what is the what is that of wrong going on with you, that you did not prepare well for the examinations and you thought that it is a minor examination? So Smaranika is a good girl, so you should have to try your best for the next examination. Okay. Okay, students, you forget that whatever you have scored, that is not a matter, because it is online test, and that God knows that in which way, that means you appeared the examination, that is not a factor, and you should not have to discuss about it. But main thing is that that means which is the longest phase of that of subphase of that of phase one. The answer will be that of you have made a lot of mistakes. Answer would be that of pachitin. And for that of oocytes, that is means the myocytes. Have you know that myocyte cells? That means cells which do that of meiosis is called myocytes. And that myocyte, for that of myocyte, basically to that of oocytes. That means we shall produce that is ova. That cells stage, diplotin stage, is for that of 13 years. It is that of longest surface for that of oocytes. But for that of common for all myocytes, that it is pachitin. Okay, pachitin is the right answer. That is, it is the longest surface of meiosis one. But if a question would be that which one is the longest surface for that of oocytes, the answer would be diplotin. Okay, the most of students have done mistakes owing to that region. They have got 0.5 marks. Okay, so don't forget it. That is not a matter. That means what you have scored or what you have not scored. Anyone scored more, anyone scored less. This is not a factor. Factor is that how you have to prepare yourself for that of your the final destiny. That is your that board examination, as well as that of NEET or any entrance examination. Okay, that is the factor. That is the matter that you must have to realize. Now, students, you come to that of chapter. That means in that photosynthesis higher plants. Yesterday that we had discussion about that of photosystems. Okay, in that photosystems, I had shown you that you see that. Suppose you see that in that photosystems, what I had done, that you see that here you see that here is the reaction center, and there are so many pigments are there. Okay, so many pigments are there. These pigments are commonly called as what? That is light harvesting complex. That is light harvesting complex. Okay, and this is that of that is reaction center. That is reaction center. Reaction center. Okay, reaction center. Reaction center. If reaction center is P700, then it will be as PS1. Okay, PS1. And why it is called PS1? Because it was discovered first. It was discovered first. Okay, discovered. PS1. One means it was discovered first, and that means wavelength. That means the light wavelength is to that of 700 nanometer. 700 nanometer. Okay, and that reaction center is always occupied by that of chlorophyll A, having that of 700 nanometer. And these are that of this part, these are the light harvesting complexes. So many pigments which supply energy and electrons to that of chlorophyll A for that of photosynthesis. Only that of reaction center students, reaction centers do photosynthesis. That is reaction center. Why it is called PS? That is photosynthesis, because that system, it has machinery, it has such mechanism which can convert that of light energy to that of chemical energy. Okay, and that are okay, it has also mechanism that can convert, that is photolysis of water. That means it can also break the water and to get the electrons. So I will tell you. So it is a what? It is a photosystem one. And in photosystem one, here we will have to see that that is primary electron acceptor. That is primary electron acceptor. it will have to accept the electron first. That means which electron will be excited from that reactor, that will be accepted first by the primary electron acceptor. Okay, acceptor. And here are that of enzymes. Okay, enzymes, presence enzymes, because reaction center must have enzymes in order to break the water, so that photosystem can get the electrons as well as can have to release the energy. That is release energy as well as also oxygen. Okay. So that photosystem, students, you have to remember this is photosystems. That is one, that is P700 nanometer. If it would be, I would have to write down here that is 680. I have to write down 680 here. It would be photosystem 2. It would be photosystem 2, and it was discovered later on. Discovered, that is, it is discovered later on. Okay, later on. So it is photosystem 2. So 680 chlorophyll A can have to accept that of that is 680 nanometer of chlorophyll present here. Going to that region, it is called that of photosystem 2. Okay, and both are cells. That is primary electron acceptor and that is of enzymes. I will tell you that is primary electron acceptor for photosystem 1 is that of APS. That is, I will have to tell you, don't be hurry, that is I will have to tell you. And for that of that is photosystem 2, that is pheophytin, I will have to tell you later on. Okay. Now you remember that this is about that of photosystem. Photosystem is a part where that photosynthesis takes place. Photosynthesis, that is photosystem is just one molecule of chlorophyll A that has mechanism which can convert light energy into that of chemical energy. So photosynthesis can only take place in that of reaction center. Okay, reaction center, and that reaction center has that of reaction center is in either in the photosystem 1 or photosystem 2 on the basis of chlorophyll wavelength of chlorophyll. That means which type of wavelength of light can chlorophyll accept, and chlorophyll possess, that is either 680 or 700. That depends upon that of photosystems. Okay, this about that of photosystem 1 and photosystem 2. And I had also students yesterday told you that photosystems present. Suppose I am drawing here. You see that I am drawing here. Is suppose think that it is that of mesophyll cells. Suppose you think that it is a chloroplast. Okay, chloroplast. You think it is a chloroplast, and this is that of outer membranes. That is outer membranes, and this is that of inner membranes. Okay, inner membranes. And here you see that is thylakoids are present. Okay, that thylakoids are present like this. Thylakoid are present here also. That of thylakoids are present. Okay, that thylakoids are present here. You see that, and these both thylakoids are joined by that of that is stromal lamellae. Okay, that is stromal lamellae. It is joined that of thylakoid, and there are some also that are not joined. And you can have to see here, students, that here you can have to see that this is called what? This is thylakoid. That is thylakoid membrane. Okay, this one. And here you can have to find that of photosystem 1. Here is photosystem 2 is present. Here is photosystem 1 is present. Here is photosystem 2 is present. Here is photosystems 1 is present. Okay. And here also you can have to find that of photosystem 1. Only photosystem 1 is present in that of stromal lamellae. Okay. That is photosystem 1. Only photosystem 1. So both photosystem 1 and photosystems 2 are present in that of thylakoid membranes. In that of thylakoid membrane, that PS1 and PS2 are present. The point is very important. Okay, that is photosystem 1 and photosystem 2 are present on that of thylakoid membrane. But only photosystem 1 in that of stomma lamella, in that of stomma lamella, okay, that what is present? Only that of photosystem 1. Okay, so photosystem 1 and photosystem 2 both are present in that of thylakoid membranes, and that of photosystem 1 is present only in that of stomma lamella. Okay, that concept should be clear. Okay, and you see that now we'll have to know about that of transport of electron. Yesterday I had told you that when that of light energy will fall on that of chlorophyll, that will be excited, or on that of any pigments of that either of chlorophyll A or B, or that of xanthophylls or carotenoids, that will be excited. So electron will come out, and that process of electron transport. Today we'll have to know about that of electron transport. Okay, that is electron transport. We'll have to know about that of electron transport. Okay, electron transport. You see that I have told you that both photosystem 1 and photosystem 2 are found in that of thylakoid membrane. Suppose I will have to, you see that I am just drawing a, you see that just once. Suppose this one is a, suppose you see that, suppose this one is a thylakoid membrane. Okay, this one is a thylakoid membrane. This one is a thylakoid membrane. And in this thylakoid membrane, you see that, students, here I have drawn that of photosystem 2. Okay, here I have drawn a photosystem 2. Here I have drawn a photosystem 1. Okay, that is PS1. Here is that of PS2. Okay, this is photosystem 2. This is photosystem 1. What I have told you that both photosystem 1 and the photosystem 2 will be excited simultaneously, not one after another. It will be excited simultaneously. So that here is the photosystem 2. So that is light energy that is coming on, and here also see that that is also light coming on, and you see that 600 nanometer, that is 600 nanometer. If light energy is more than that of 600 nanometer, that means the two photosystem will not be excited. Okay, that concept should be clear. If photosystem, if that of light, that is light energy that is coming, okay, that light is of more than 680 nanometer. That means that PS2 will not be excited, because it will not have to receive that of light energy. Okay, that light energy cannot be received by that of photosystem. So less than, so must be less than that of, so that which will have to accept. That means that is light harvesting systems. You see that suppose it is a reaction center, and that both all that of reaction center is encircled. The reaction center is encircled by that of light harvesting complex. So there are many more pigments are there. Okay, here is that of this one is that of a reaction center, but these are that of light harvesting complex. So they will have to receive less than 680 nanometers wavelengths of light. Okay, which one? That light harvesting complex. That it is PS2 can accept maximum to that of 680. So this light harvesting complex, because you know that means 800 nanometer, 1000 nanometer also can have to. But these light harvesting complex, they can have to accept, but they can have to transfer that. They cannot transfer such type of wavelength of light to that of reaction center, because reaction center can accept up to 680 nanometer wavelength of light. If it will have to receive maximum, then it will be what? It will defunction. That means it will not function. Okay, that means photosynthesis will immediately stop. Reaction center can accept photosystem to reaction center can accept that is wave of light up to that of 680 nanometer, not more than that. If more than that will be given, then it will be automatically break down. That means photosystem will automatically break down and the process of photosynthesis will immediately be stopped. So less than, so less than, so less than 680 nanometer will have to be received here. Okay, received here, not more than. More than 680 nanometer is not acceptable. Only 680 or that less than 680 nanometers are acceptable. So it is accepted. So that you see that here what happens, students. Here what happens. That means here electron excited. Okay, here electron excited. Okay, electron excited, and that electron which one excited, that one will be accepted by a primary, that is electron acceptor. Okay, that is primary electron acceptor. So here is a primary electron acceptor. Okay, that means primary electron acceptor is one which can have to receive, which can have to accept the first electron first of all coming from that of systems, from that of reaction center. Okay, so that electrons comes and it is received by what? That is received by primary electron acceptor. This one is called primary electron acceptor. Primary PEA. PEA stands for PEA. PEA stands for primary electron acceptor. Okay, very good concept, students. You must have to remember. And the PS2 primary electron acceptor is called phosphatin. Okay, this one's name is phosphatin. I'm just writing here, students. That is phosphatin. This one is phosphatin. Okay, phosphatin. So primary electron acceptor of primary, if you see that, that means electron which excited and comes out of that of reaction center may also chance of also returning to that of reaction center. But it is accepted, but if it does not go to that of original place, that means it will have to accept by a primary electron acceptor, that is phosphatin. Phosphatin. Okay, phosphatin is the first electron acceptor. That means it will accept. But after accept, that means it will have to donate another primary electron carrier. There are several electron carriers in that system. In PS system, there are several electron carriers. Okay, which can have to accept such electrons. Okay, electrons which have been immediately accepted, that will be oxidized, and whenever that will, after that it will be reduced form in reduced forms. So that will have to be given, and this one will be given to another one. This is another. Okay, you see that this is a electron carrier. This one is a electron carrier. There is another electron carrier here also, students. Okay, here also another electron carrier. And the electron also, that means electron which are in reduced form, they will move downhill. They will move what? That is downhill. They will move. Okay, they will move to that of downhill. Okay, they will move to that of downhill. They will move down. In the downwards position, they will have to reduce. Okay. So that means you see that is electron acceptor. So the PS2 electron acceptor is known as phosphatin. After phosphatin, okay, it will have to give to that of another one, students. Okay, another one that is called plastoquinone. This one is called that of this one is called that of I'm writing, that is writing here, that is called this one. This one is called this. It will give to this one. This one is 1, this one is 2, this one is 3, this one is 4. So that this two is known as plastoquinone. This was known as plastoquinone. Okay, quinone. Shortly, you can have to write down PQ. Okay, you can have to write PQ. That is plastoquinone. quinone. So that is plastoquinone. And the plastoquinone will have to give to that of that is cytochrome. Okay, cytochromes. It will have to give to that of cytochrome C, that is B6F. Okay, that will have to cytochrome B6F. It will have to give to that of cytochromes. Cytochrome B, that is B6F. Okay, B6F. And then it will have to go to that of plastocyanin. Okay, then it will have to plastocyanin. Then it will go to that of plastocyanin. Okay, that shortly known as PC. It is also shortly known as Ci2B6F. Okay, it is shortly known as PC. It is PQ. So that you see that means less than 600 nanometer light when that falls on that that is reaction center PS2. That means electrons are excited and that electrons come out from outside, that come out of the center, and it will be accepted first of all by that of a primary electron acceptor. That primary electron acceptor will give to that of electron carrier. There are 3 electron carrier, and these 3 electron carrier are known as that of that is electron transport chain. That means electron transport chain. That is electron transport chain is made up of 3 electron carriers. Which are these 3 electron carrier? First one is that, students. That means that is electron transport chain is made up of that of 3 electron carriers. Which are these 3 electron carrier? One is that of plastoquinone. Second one is cytochromes B6A. Third one is that of plastocyanins. So that is PC. PQ, B6A or PC, you can have to tell shortly. So this one, that is electron transport chain. So your thiophytin, which is the primary electron acceptor, it will accept the electrons and it will give to that of electron transport chain. Electron transport chain of photosystem 2 is made up of 3 electron carriers. Which are these 3 electron carriers, students? First one is
plastoquinine.
Yes, plastoquinone. Then second one is that of
Cytochrome.
cytochrome
B6F.
B, that is
6F.
6A, B6A, cytochrome. Then it will have to give to that of
Plastocyanine.
plastocyanins, that is PC. So these are the electron carriers. These electron carriers, they will have to take the electrons from that of thiophytin, from plastoquinone to that of cytochrome to that of plastocyanin. This is called electron transport chain. You have to remember. And whenever it will reach that of cytochromes, it will have to starting the preparation of ATP. That means ATP synthesis will be occurred here, students. That means where ATP synthesis here, that is ATP synthesis from that of ADP and that of phosphate. Okay, phosphate. That means ATP will produce. ATP will produce. ATP will produce when electrons reach the cytochrome B6F. There you can have to produce that of ATP. So ATP will be produced from ADP and phosphate. From ADP and phosphate, that ATP will be produced. Okay, that is adenosine triphosphate. That is adenosine triphosphate will start producing at which place? That is cytochrome. Cytochrome B6A. When electron is at that carrier of cytochrome, is at the place of carrier of cytochrome B6F, at that time, that is ATP production will start. ATP production will be started at that time. So then it will have to go to that of then electrons at a reduced form will go to that of plastocyanins, and from that of plastocyanins, that it will have to move to that of PS1. I have told you that that means electrons. That means always that electrons. That means one chlorophyll. That means one pigments will donate electrons to it is higher wavelength. Okay, that means a pigment of a lower wavelength will donate it is will give it is electron to that of a higher wavelength. So that you see that here is wavelength is 700, here was 680. That means these electrons were given to that PS1. So that PS1 get that electrons and that will be excited, and sunlight at the same time sunlight also falls. It will be excited. That electrons also come out. Okay, and these electrons will come out. Which one? That electrons will come out. That electrons will come out. That electrons will come out, and that electrons will accepted by a primary acceptor that is called APS complex. Okay, it is a primary acceptor. It will have to receive that this is the primary acceptor of that PS1, that is APS. APS complex is that of a primary acceptor of electrons. Okay, electrons. Primary acceptor electrons of PS1, that is APS complex. It will accept and it will have to give to whom? That is okay, ferroductins. It will give to that of ferroductins. Okay, ferroductins. And that ferroductins, that is FD. Shortly it is called as that of FD. Okay, FD. That ferroductins, that means it will the compound these ferroductins. That means it will converted what? It will convert that of it will reduce that of NADP plus to that of NADPH plus H plus. That means when you have to mark it that plus sign, it is in reduced forms. Okay, that means when NADP only written NADP, you have to add here what? You have to add here one hydrogen, one electrons, and it will be NADPH. Okay, NADPH. But when you see that, whenever we see that NADP plus is marked with the plus sign, you'll have to see that it is in reduced forms. It is in reduced forms. It is in reduced forms. So what it needs? It needs that of 2 hydrogen plus 2 electrons. Okay, that is 2 protons and 2 electrons it needs. So that after that it will become what? It will be NADPH plus H plus. Okay, so you see that if you will have to NADP only, that means it requires. That means it is not in the reduced stage. That means it requires only 1 hydrogen and then that is one proton and 1 electrons, and that will be NADPH. But if it is plus sign given, if you have to mark that NADP plus, what is plus? That is in reduced form. In reduced form means you have to add that of 2 protons with that of 2 electrons. So 2 protons and 2 neutrons must be added in that of reduced form, so that in order to be reduced, so that means you have to add. If you have to add protons, if you have to add the protons, then you'll have to reduce it into that of NADPH plus H plus. That means it is the reduced form. It is the reduced form of NADP. NADP reduced form is NADPH. If it is plus sign given, then it is in reduced state. So you should have to add 2 protons and 2 electrons. So you should have to get NADPH plus H plus. Okay, and it is being and it is catalyzed by that of an enzyme present there. That enzyme is called ferrodoxin. That is ferrodoxins and NADP reductase. That enzyme's name is ferrodoxins. Okay, ferrodoxin NADP reductase. Yes. Okay, reductase. That means which enzyme is involved in the process of converting, that is reducing NADP to NADPH, that is ferrodoxin ferroduxin NADP reductase. Okay, NADP reductase. This enzymes that is which is present in that of compound of ferroduxins. It is function is that of to convert NADP to that of a NADPS. So one of the product that means you have get in that of ATP, another product in that of light reaction. This photosystem are part of light reaction. So in the right reactions you have received that of ATP at which site, at which that of that is cytochrome B6F. Okay, cytochrome B6F, that ATP and phosphate convert into that of ATP. And here also in that photosystem once, that means when electron excited and come out, it is accepted by that of a primary acceptor of electrons, that is APS. That APS will have to give to that of a ferroduxin in that of reduced stage. And that ferroduxins, that in ferroduxin, that ferroduxin NADP reductase enzymes are present. And that enzymes will immediately convert NADP to that of a NADPH because it gets the electrons. So that of that is chemical reactions will occur. That is a reduction process will occur because electrons has received it by ferroduxin. So it will also be, that means it will be activated and it will start reducing NADP to that of a NADPH H plus. Okay, if it is have to be marked with that of a plus sign. NADP plus means it is reduced forms, and it will have to added with that of 2 hydrogen and 2 electrons, 2 protons and 2 electrons. But in case of NADP only present, not given plus sign, you have to add 1 electron with 1 proton with that of 1 electron. So you have to get NADPH. Okay, students. So you see that. That means when you see that, it is very important. So you have named the process. I have already told you that. I'm just asking you that questions. You just have to answer me. What you have understood about that of transport of electrons. First of all, that in PS2, that means when, that means it gets that of light energy of 680 nanometer or less than that. That means that reaction center excited, and that means electrons comes out, and that electrons from that of PS2 will be accepted by that primary electron acceptor. That name the primary electron acceptor of that PS2. Yes.
518.
518. Very good. Then come to that after that 518. That means it has go. That means it will have to be accepted by electron carrier. That means which is the first electron carrier that will have to accept the electrons. This one is yes. Then next one is
Cytochrome.
Cytochrome B6F, and that means where ATP, that means ATP synthesis will take place. At cytochrome B6F. Okay, at that center, at that place, that means it will have to ADP will have to be converted into that of ATP. And after that that means after cytochromes, electron will move to that of yes, and after that it will go to that of
PS1.
PS complex. Sorry, PS1.
PS1, that means electrons, that means chemical reaction, process of chemical reaction starts. So that electron will have to come out. That means excited. That means electron will come out. Which one is the first electron acceptor?
FES complex.
Yes, that is FES complex. And FES complex will have to donate. Will have to give electrons to which compound? That is ferroduxin. In ferroduxin, which enzyme is present?
Ferroduxins are an NADP
Ferroduxin, that is NADP reductase. And what it will convert? What it will have to reduce?
NADP plus and NADPH plus H plus.
NADP plus will have to be converted into, that will be reduced to that of a NADPH plus H plus. Very good, brilliant answer. Okay, and you see that, and mindfully you see that. That means when PS2 donate electrons, that means it will have to the less amount of electrons. So that means PS2 will have less amount of electrons because if it gives electrons, whatever electrons be excited, it gives to that of electron carriers and they are taking it, and that photosystem too will be of deficit of electrons. That means photosystem too will have to fill that of a deficit of electron. Will face the problem of deficit of electrons. Then what it will do? To that get the electrons immediately, quickly get the electrons. So that means that is photolysis of water will occur. So it will break the water. Okay, that photolysis of water it takes place. That means if water breaking, lysis means what? That a breaking of water. So breaking of water. So PS2. So photolysis of water takes place in that of PS2, not in PS1. It is very important concept, students. You must have to remember. That is photolysis of water that is present in that of PS1. Sorry, PS2, not in PS1. That is photolysis of water is not present in PS1. That is present in that of PS2. Okay, PS2. That is second PS. That is it is found that of photolysis of water. That means when PS2 faces that of what? PS2 that is face that of deficit of water. Immediately it will break water. Okay, that water will be disintegrated. So if that means water disintegrated, what you will have to get? That is 2 hydrogens, that is 2 protons you will have to get. Then we'll have to get that of O2. Then we'll have to get also electrons. That electrons will move to that of photosystems. So immediately deficit of electrons being compensated by that of photolysis of water, that is breaking of water. Okay, and in that breaking of water, that is enzymes which takes part in that of that is called water oxidase. That is called water oxidase. The enzyme, that is the enzyme which is involved in the process of that is breaking of that of water into that of hydrogen and that of oxygen and electron. That is water oxidase. And this process is also held by that of that is ions, that is 3 ions. 3 ions that is help in such process. They are manganese. Okay, manganese, then chlorine and that of calcium. That is manganese, chlorine. It is very important, students. Which that means which mineral ions take part in that photolysis of water? Answer will be which 3
Manganese,
mineral ions?
manganese, chlorine, calcium.
Manganese, chlorine, and then calcium. It is very important. Manganese, chlorine, and calcium. And which enzymes takes part? That is water oxidase. Okay, this one is called what? Water oxidase or water splitting complex. It is otherwise known as water splitting complex. That complex also the term related to that of enzymes. So that water splitting complex or water oxidase. This enzyme it is. So that breaking of water into that of hydrogens and that of oxygen and electrons is that of a enzymatic process. Enzymes, that is water oxidase enzymes takes part in such breaking. So and it is supported by that of mineral ions of manganese, chlorine, and calcium. These 3 are very important. You must have to be remembered. And that of photolysis of water, if you have to make that of 2 hydrogen, if more electrons are needed, so more molecules of water will be break. So in this case also we'll have to get 4 hydrogens for that of 2O. Okay, 2O and that of that is electrons, 2 electrons. That is 2 electrons also you have to get. So that more, that means more electrons needed mean more molecules of water will be Okay, so that in that case we will have to get that of four, that is four protons, that is four, that is oxygen also, and that oxygen, students, that oxygen what you will have to get, that is very important one. That is this oxygen is needed for that of a respiratory process. Okay, this is for molecular oxygen, that what you will have to get, that is needed for that of a respiratory process, and in that of respiration, that is needed. So it is released to that of outside to the atmosphere, electrons being given to that of photosystem 2 to compensate the loss of electron, the deficit of electrons. When that photosystem 2 face the deficit of electrons, immediately water, it starts breaking water. So by breaking water, it will have to get that of electrons. That electrons will have to immediately go, and that hydrogen will reach in that of that means stroma. Okay, stroma. In that of that is in that of chloroplast. You know that the middle of that of chloroplast is that matrix of stroma. That hydrogen, that protons will reach in that of matrix stroma, and that oxygen will come out into reach that of atmosphere, and here is that of electrons that will compensate the loss of electrons in photosystem 2. So photolysis of water is found in photosystem 1 or photosystem 2?
Photosystem 2.
Yes, in photosystem 2. It is found in photosystem 2. Okay. And that is the process of that of electron transport. And it is very important, students. You must have to remember. Okay. You must have to remember that one. It is very important. And you see that. And that means what I have told you, that protons, I have already told you that protons are likely to be released to the liquid. That means this hydrogen, that means these protons will to be released to that of a lumen of thylakoid, and that is lumen of the thylakoid, and also that of the stroma of matrix of stroma, and this one will go to that of thylakoid membranes, will come out to the thylakoid membrane and go to that of atmosphere, and electrons will have to reach to that of photosystem 2. Okay, this is the process of photolysis of water, and that of photosystem 2 and photosystem 1, how they work simultaneously, and how electron transport in that of photosystems. And that electron transport chain consists of that of that is plastoquinone, and that of cytochromes, and that of plastocyanins. That we must have to remember. This is very, very important. Okay, this is very important. You must have to be remembered. And then we will have to know about that of photophosphorylation. Okay, photophosphorylation. This is also very important. There are two types of photophosphorylations. I'm just telling one about another. Okay, that is, you see that is photophosphorylations. In that that means you know that living organisms have the capability extracting energy from oxidizable substances. Okay, living organisms have capability of getting that of energy from that of oxidizable substances, and that energy will have to deposited in the form of a bond, that is ATP. That is one example of that of bond energy, that of ATP. So that what you have understood, that means living organisms will have to receive energy from that of what, oxidizable substances, and that energy will be stored in the form of a bond, in the form of a bond. In that of what I have told you, that in photosystem 1 and that of photosystem 2, you see that means it is a process is going on, that is process is going on. And if that means photophosphorylation, in that of I am just telling you that is photophosphorylation. Okay, that is photophosphorylation. You understand that? That means phosphorylation means that is ATP synthesis. Okay, that is synthesis of ATP. This understand. This part you understand, that phosphorylation means synthesis of ATP. ATP synthesis is called phosphorylation. When ATP is being synthesized in chloroplast with the help of light energy, it is called photophosphorylation. So photophosphorylation is related to synthesis of dash. Synthesis of dash.
ATP.
ATP. So that phosphorylation means synthesis of ATP. But and you know that ATP is synthesized in two organelles. One is that of mitochondria. Another one is that of chloroplast. Okay, in chloroplast. That is in animal body, that is it is in mitochondria, and in that of plant body, it is in that of chloroplast. So there are two organelles where ATP is synthesized. One is mitochondria, two is that of chloroplast. That is synthesis of ATP in mitochondria, student, is called oxidative phosphorylation. That is relations. In that of synthesis of ATP in mitochondria is called oxidative. Okay, oxidative phosphorylation. Okay, photophosphorylation. You must have to understand that phosphorylation means synthesis of ATP, and ATP is synthesized in two organelles. One is mitochondria, another one is chloroplast. Mitochondria in animal cell and chloroplast in that of plant cells. And synthesis of ATP in mitochondria is known as a new name, that is oxidative photophosphorylation. But when it comes to that of chloroplast, it is known as what? It is known as photophosphorylation. That is concept is clear. Have you understood that concept? That is,
Yes, sir.
that is phosphorylation means what?
Synthesis of
Synthesis
ATP
of
in
ATP.
chloroplast.
ATP synthesis, and ATP is being synthesized by two organelles. Which are these two organelles?
Mitochondria
and chloroplast.
And chloroplast. Mitochondria is found in that means ATP synthesis in animal cell in mitochondria, and that process is called
Oxidation.
oxidative phosphorylation, not photo. Oxidative. Oh, I am written wrong. That is oxidative phosphorylation. Okay, oxidative phosphorylation, not photo. Photo means light, that is chloroplast. Okay, oxidative phosphorylation. And when synthesis of ATP is occurred in chloroplast, it is known as
Photophosphorylation.
photophosphorylation. Understood? In mitochondria, ATP synthesis process is called
Oxidative phosphorylation.
oxidative phosphorylation. Oxidative phosphorylation. And when it is occurred in chloroplast, it is known as photophosphorylation. Okay, oxidative phosphorylation. Don't tell photo. Okay, oxidative phosphorylation in mitochondria, and in chloroplast it is photophosphorylation. This concept must be important, and what you will have to read about that of photophosphorylation. You don't have to read about that of oxidative phosphorylation, because you know that oxidative phosphorylation is that process is found in animal cell, and it is being held in that of mitochondria, that is ATP synthesis. And what you will have to discuss, that is about photophosphorylations. Okay, what you will have to discuss, that is photophosphorylations. Photophosphorylation. That photophosphorylation, students, is of two types. One is that of a cyclic. And another one is that of a non-cyclic. It is very, very important, students. Okay, photophosphorylation, synthesis of ATP in chloroplast is called photophosphorylation. It is divided into that of two types. One is cyclic, another one is non-cyclic. Another one is student non-cyclic. One is cyclic, another one is non-cyclic. Okay. Now I am going to discuss about that cyclic photophosphorylation. Now I am going to discuss about that photophosphorylation, cyclic photophosphorylation. Then I will have to discuss about non-cyclic photophosphorylations. Okay, that is cyclic photophosphorylation, non-cyclic photophosphorylations. Okay, first we will have to discuss about that cyclic photophosphorylations. You see about that. Okay, you see that. Students, you see that cyclic means how it is in circular forms. You see that. Suppose it is that of reaction center, and these are that of what? These are that of these are that of pigments. Okay, these are that of pigments. These are that of pigments. Here you see that sun, that is sun, that is sun. And you see that means light waves comes, and that is, you see that is PS. Okay, that is PS1. Okay, photosystem 1. So photosystem 1, that is reaction center. So that light, that is light energy falls on that photosystem reaction center. So that reaction center, in that reaction center, electron excited, and that electron will come out. Okay, and you see that electrons come out, and that electrons will first accepted by that is APS complex, and it is being accepted by that APS complex. Okay, it is accepted by that. It is the primary electron acceptor. Okay, and after that it will have to go to that where it will have to go. Okay, it will have to go to that peroductions. Okay, that peroductions. Okay, in that peroduction, I have told you that is NADP will be converted into that okay, into that what? It will be converted into that NADPS. After that, it will come to that that is cytochrome. Okay, that is cytochrome B6F. Okay, that is cytochrome B6F. And then it will come to that plastocyanins. It will come to that plastocyanins. Okay, that will plastocyanins. It is come to that plastocyanin. Then after that, it will have to return back to its original place. So this one is called that cyclic photophosphorylations. Okay, this one is called cyclic photophosphorylations. It is called cyclic photophosphorylations, in which electron excited and it is accepted by that primary electron acceptor, that is APS complex. Then it gives electrons to that peroductions, and this peroduction also, in that peroductions, that we know that is NADP is converted into that NADPH plus. Okay, so that is reduction takes place NADP to that of NADPS. Have you students remembered full form of NADP? Have you remembered? Anyone can tell?
Sir, nicotine,
Yes.
diphosphate.
No. You see that I'm writing nicotine. Okay, nicotine amide. Okay, nicotinamide, adenine, dinucleotide, phosphate. In that of enzyme chapter, I have already told about it. You have to remember. It is your turn. You must have to remember. I'll have to just tell. Clear your doubt. Build up your concept. But on your part, you can have to remember. If you have to forget, then what can I do? That is nicotinamide stands for N. Then adenine, that is A. Dinucleotide, that is D. Then PH, that is phosphate. Okay, that is both NADP and NADPH. That is the common name nicotinamide, adenine dinucleotide phosphate. That is common for NADP and NADPH because NADPH is a reduced form of NADP, but both are same. So nicotinamide, adenine dinucleotide phosphate. Okay, so that is NADP. So this is the cyclic photophosphorylation. You see that? That means when sunlight falls on that of reaction center, that means on that of light harvesting complex. These are the light harvesting complex, how it is giving protection to reaction center. Okay, they will die, but their master will not die. Okay, so that means this they give to that of electrons to PS1. So PS1 excited, that means it gives electrons to PSS, that is the primary electron acceptor. That electron being received by ferrodoxins. Here NADP reduced to that of NADPH. Then electrons comes and reach that of cytochrome B6 complex, B6F, and then it is given to that of plastocyanins. Then it is returned back to that of PS1, that is reaction center. So where it has come, it is again returned back to that place. So this is called that of cyclic photophosphorylation. So this is a cyclic. You see that this is a circle one. This is a cycle one. Going to that one, this is called cyclic photophosphorylation. And in that that means today's, that is evening class. So today's Wednesday, you might have remembered. And today in the evening class, I will have to teach you about that of non-cyclic photophosphorylations and that of chemi-osmotic process in which that of ATP synthesis, how that of mechanism, the process of, you know about that of NADPH reduced to that of NADPH, you know that, but that is ATP synthesis which takes place in that of, that is, you must have to know it in that of evening times. Okay, at the evening time, evening class, I will have to discuss about that of non-cyclic photophosphorylations, difference between the two, and then I'll have to chemi-osmotic process. Okay, then I'll have to finish that of light reactions, and from tomorrow is a holiday, and the day after tomorrow that I'll have to start with that of dark reactions. Okay, dark reaction. Now students, I'll have to ask only 2 questions and then we'll have to go. Okay, the questions are now ready to write out. The first questions, okay, which of the following statement is incorrect regarding crossing over? Okay, regarding crossing over. You have to write down the questions. Select the incorrect statement regarding crossing over. Question 1. Select the incorrect statement. Incorrect statement regarding crossing over. Okay, I'm giving you to that 4 options. It occurs between sister chromatid of the homologous chromosomes. It occurs between, okay, it occurs between sister chromatids of homologous chromosomes. Between sister chromatids of homologous chromosomes. First point. First, that is option. Second option, it is the exchange of genetic material. It is the exchange of genetic material. Okay, it is the exchange of genetic material between two homologous chromosomes. Shortly writing. Okay. Option C is that it is an enzyme mediated process. It is an enzyme mediated process. And option D, student, is that it leads to recombination of genetic material. It leads to recombination of genetic material. Okay, genetic material on the two chromosomes. On the two chromosomes. Can students give the right answer? Anyone? Yes, Anamika A. Yes, Smaranika also right answer. Smaranika right. Shanvi, no, you are wrong. That is Adhyasa Dubey, right answer. Tushar Ranjan, wrong answer. Jyoti, right answer. Mona Lisa, right. Satyaprakash, right answer. Avinash, right answer. Satyaprakash. Beula Khora also right answer. Okay, Beula Khora. Priyanka Bag also right answer. Okay, right answer. You see that, students? I am telling, very bad indeed. Who has given the option C? How is it? Crossing over is completely an enzyme mediated process. Can you tell me three enzymes that are related in that crossing over? Anyone? Anamika?
Sir, endonuclease and ligase.
Yes, endonuclease and ligase. But what is in your NCERT book you have forgotten? That is anyone?
Recombinase.
Yes, recombinase. So it is an enzyme mediated process. Three enzymes take part. Three enzymes. One is recombinase. It is given in your NCERT book. But I have told you two enzymes extra. One is ligase. Another one is nuclease. Okay. Endonuclease. Okay. That is endonuclease. That is this enzyme. Recombinase, ligase, and endonuclease. These are related to that of an enzyme mediated process. And it is the exchange of genetic material between two homologous chromosomes. True. It is true. It leads to recombination. That means you are looking different. Our father, mother are of one type, but you are not either a father or mother. It is because of that of genetic material recombinations. You are looking different. You are not alike either to our father or to our mother. You are in a separate, that is different complexion or that of different shape, different face of our body. It is due to that of that is genetic, that is different also talent. You are also varies from either you more than father or less than father. That is recom. So it is also true. But it occurs between sister chromatids of homologous. That will be non-sister. I am just drawing, you see that. Here is that of one chromosome. Here is that of another chromosomes. These chromosomes and this is, okay, you see that. That means this chromosome pair with. That means this one is a, this one, two are sister chromatids. But 3, 4 are also sister chromatid. But 2, 3 are, these 2, 3 are non-sister. Okay, these 2, 3 are non-sisters. Okay, you have to remember. 1, 2, you see that. This is a chromatid. This is another chromatid. This chromatid, are you seeing that one? Not visible. Okay, I am drawing here. You see that. This is one chromosome. This is another chromosome. This is chromosomes, that is chromatid. This one chromatid. This is another chromatid too. This is chromatid 3. This one is chromatid 3. This one is chromatid 4. Okay. And you see that 1, 2 relation, sister chromatid. 3, 4 relation, 1, 2 relation, sister chromatid. 2, 3, 4 relation also sister. But what is the relation between 2 and 3? 2 and 3?
Non-sister.
Non-sister. 2 and 3 non-sister. Non-sister. You see that? This, you see that? And here also, you see that 1, 3 relation also, 1, 3 relation also, non-sister.
Non-sister.
This term, this one also, that means that you see that. That means how one is made. This is called what? Recombination nodules. Okay, this one is a recombination nodules. This one also recombination nodules. This one also recombination. You see that this is the point of crossing over. These recombination nodules are the points where crossing over takes place. So crossing over occurs between two non-sister chromatid. I have given question sister chromatid. How it is in between sister chromatid? If sister chromatid, then that means all will be that of equal appearance, equal face, equal shape, equal genetic material, everything will be same. So that is between two non-sister chromatids. Okay, very careful about it. Very important questions for examination point of view. Okay, students, that is time is on, and in the evening sessions I will have to do that of 5 questions. Okay, in the evening session I will complete that of non-photophosphorylation, compare between photophosphorylation, cyclic, non-cyclic. Then I will have to chemiosmotic process, that is synthesis, that is process of synthesis of ATP. I will finish that one. Then I will have to ask you 5 questions. Okay, questions what I have taught in that of chapter of photosynthesis, as well as that of also that of cell cycle. And I am planning that from Friday onward, I will tell in the evening time that we will have to remember only 3 pages of that of bio books. That process we should have to continue till the school is, that is till our school is open. You will have to continue such process. Only 3 pages, not more than that. 2 or 3 pages. We'll have to decide. We'll have to tell me that whether you'll have to remember 2 pages or that of 3 pages. Every day, 2 pages, 3 pages, I'll have to make 3 questions. 3 questions. If 2 pages, I'll have to make 3 questions. If 3 pages, I'll have to make 5 questions. And that questions, I'll have to make questions and will ask you on the whiteboard, and you'll have to answer it. Okay. Okay, students, by this, I'm finishing that today's class. Okay. Thank you very much for joining.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you. Thank you.